Great. So where are we? We have, over the last few days, we've introduced ourselves to Bioconductor, have all these requirements. If we have problems, we're asking help and get help. We introduced ourselves to what is Bioconductor, this framework for handling biological data in R. So great for us and what we want to do. We introduced sequences in Bioconductor. How do we hold sequences in general, really from FASTA files to Bioconductor? And this allows us to handle things like genome builds, our reference genomes. We also saw that we have these BS genome packages, and these are great because they allow us to just pull the sequence we need for alignment or for interrogating our genome straight from a Bioconductor package. And then we looked at this real fundamental type of objects inside our Bioconductor infrastructure, and that is actually Genomic Ranges package, Genomic Ranges. Okay. I seem to focus on calling them genomic intervals though. So this is going to allow us really to handle anything which has a chromosome start end. Most genomic features can be represented like this. And we saw how we might be able to manipulate them, modify these, export them, and import them from bed files. Okay, so this guy started to get us going on just how we start to work with kind of genomic positions, genomic intervals. So then just to add to that, we have genomic intervals with some type of an association of a score. And these become our genomic scores. And often we will see these in things like beg graphs and bigwig files. This is what we want to look at in IGV, this kind of graph track. Also wiggle files, because they're a wiggle, right? They're a graph. We saw that we can input these from pre-compiled scores, things like bigwigs and beg graphs. We can input them, we can manipulate them. We can actually create these RLEs in R, right? These run length encoded values, which represent scores across the genome. And then we saw that we could get some type of summary statistics from these by using these bigwigs, beg graphs, importing them. We could find the mean signal over particular regions, and that was reasonably useful, giving us, our strongest peak, most enriched region. And we saw in the exercise that that correlated really well with what they're pulling out of the actual peak caller they used. Okay, so we went from genomic intervals to genomic intervals with some values attached. And then we started to look at genomic features. This is how we hold gene models. So they're in GTF, GFF, and then we took our TXDB packages, and that contains the gene models as well, right? So pre-compiled gene models for us. But we saw that it doesn't really matter. We can use a GTF, GFF, we can import, export to this, and we can create our own TXDB objects. And we can use all the functionality in Bioconductor for that. We also saw how we add annotation, which is really important. How do I change my ID into something I can read, my PI can read? And then today we started to work with the actual data we've got, no longer for structures, but actually taking the data, FASTQ, our raw sequence data. How do we get this into R? How do we do a little bit of quality assessment? And if we wanted to, and we saw in the last session we don't need to, we can do some trimming to remove adapters or poor quality tails. Okay. So then we started to get a handle on the data, and we saw that we could use things like R bowtie 2 and R subread to align to the genome. And with things like subjunc, we could take our FASTQ and actually map it to the genomic positions and produce these BAM files, which we then saw we can import and export from R and manipulate using the RSAMTools package. But actually, if we want to get them in and do some low-level interactive manipulation, we use the genomic alignments package. And as we saw, all these alignments are handled very similarly to genomic intervals, the G ranges. So the last thing we were going to do in our session, last thing, is how do we take these BAM files and efficiently, these aligned data, and officially try and summarize the alignment signal over genomic regions. And it's not quite as straightforward because of the complexity of reads spanning regions or needing to sum up across regions in clever ways. So Bioconductor has a lot of this built in. Okay, so summarizing scores. So just talked about this. This is our raw format, our ID, our read sequence, and our ASCII encoded spread quality scores. Remember, hash is really bad. We saw that we can actually take our reference genome, our FASTA, take those FASTQs, we index the reference genome, we use our aligner, subjunct, or whatever you really want to use. Our HiSat2 is good. And we produce a BAM file, hopefully, but BAM file is just the computational equivalent of a SAM file. And this read we would have here will get turned into a aligned line here. And it contains all the same information, but importantly, chromosome, start, and how it's aligned to the genome, the cigar string here. What we want to do once we've got our data in something aligned is we often want to summarize this such that we can start to get some idea of the quantification of the signal over specific genomic locations. A lot of the time, for a lot of you, this is going to be summarizing the amount of signal I have over genes. Okay, so here we can see the complexity of this. These are our reads. We looked a little bit in IGV in this, but these look like a lot of reads, but they're not as much as compared to those which are falling over this. It's just gone over the end of the screen. But you can see we have splices, we have these lines, a read start here, ends here. Do I assign it to that exon, this exon? In RNA-seq, typically we want to assess the abundance of transcripts genes under differing conditions by assessing the number of reads aligning to a transcript's genes or exons. So for RNA-seq, we love to see something like this, where in this sample, lots of reads mapped to this gene, and in this sample, lots of reads mapped to this gene. In IGV, we could look at that and say, okay, that's definitely an interesting differential happening there. ChIP-seq, we need to do something similar. We want to take windows across the genome. And we may want to compare the number of fragments coming down from a particular IP. I formalin fix my DNA, fragment it. I have an IP for a particular protein. I want to bring down the DNA attached to that protein, map that back to the genome, and I can see where the protein was sitting on the genome. Okay. To do that, I basically map the fragments, the reads coming back in my experiment. And I try and find locations like this, where I have lots of signal in a window and very low signal in the surrounding area. Okay, so summarizing using these tools to summarize our reads and windows over exons and genes is really important for all sorts of analysis, not just bulk, but also single cell. So we can provide summarization of aligned data per base pair or per bin, as we define, using these wiggle, beggraph, or bigwig files. And this is what we've seen a lot of as we've gone through. Here you could look at this, and maybe there's a little bit more signal here, a bit more here. Often, bigwigs aren't quite good enough. They are condensed, they're strandless, remember. We've lost some information about, maybe there's a read which started here and went to there. So actually, a lot of the time, we're going to work with BAM files when we need to do summarization. Okay. Bigwigs are great for visualization, can be used for summarization, and we're going to show some of this now, but usually it's BAM files you really want to use when you're summarizing your peaks, your enriched locations from your ChIP-seq, or you're doing your counting over genes. So we're going to look at two main functions, two super cool, useful functions, the coverage function and the summarize overlaps function from the genomic alignments package. So it's not quite true. We're going to use the method from the genomic alignments package, but the coverage function exists for G ranges and I ranges. So you'll see that, right? But wherever we got it from, we're going to use this function, and we're going to use the summarize overlaps function. Coverage is going to give us per base pair signal, the scores, the alignment over the genome, right? So what we read in from a bigwig, we can actually create this internal structure, the already list, using this coverage function. Summarize overlaps, we're going to use to count, right, the signal alignments within a genomic region, but it's going to take into account multi-mapping, spanning exons. All this will be taken into account in summarize overlaps. Okay. In this session, we're going to use some aligned data. We're going to use the same data we actually visualized in the IGV session. So they had the liver body map data and the heart body map data. So we went and downloaded that from internally from their examples, so we can have the same as you would have viewed in IGV. It's also a really nice example because it's got a great splicing site in it. We're going to use the genomic alignments package, so we're going to load that first, library genomic alignments, then all these functions like summarize overlaps, coverage, they're available to us once we've loaded this library. So the coverage function can accept a BAM file path and will return this RLE list we already saw in the genomic scores lecture workshop of the number of reads at every base pair. Okay, so before we do anything, we know we need BAM files sorted. We need them indexed and to allow us to do quick operations on them. So here we just pass our sort BAM. So we're just going to pass our heart.bodymap.bam. We're going to create a file called heart.bam, but we don't give it a .bam. Remember, this just wants the prefix. And like with once we've sorted any file, we want to index it here. Any BAM file, we want to index it. And that means we can do this random access. We can retrieve parts of the BAM file rather than having to read the whole BAM file. Okay. By default, sorting by coordinate. We showed you in the last session, you can sort by query name, and you can also control the memory here if you really need it. Okay, so once we have our heart.bam in our directory, we can just run coverage function, give it the heart.bam, and it's going to go read that BAM file and translate that into signal per base pair. And it's going to return to us, hopefully something we've seen and we are comfortable with. Heart coverage, a simple RLE list. It's the RLE list we saw in genomic scores from the iRanges package. Okay. We've seen these before, and we've seen that these are a really effective way of compressing our vectors to carry these scores. Especially if you're going to have, genomes are potentially very long. We need to make sure that we're going to try and compress this into as little space as possible. So here we can see only the scores from chromosome 12 because there was only chromosome 12 in this BAM file. But it's still an RLE list, and we would, if we had a full BAM file, have an index for every chromosome. And inside here, you can see it's mostly stretches of zeros. But we have, for 50 base pairs, a stretch of 2 reads, probably mapping exactly to their place. 35, we have 3. 38, we have 1. Okay, but we've gone from a BAM file to that RLE list in just one step. We know now that we can export this if we wanted to things like bigwigs and then visualize these in IGV. Right? So already we don't have to visualize the BAM anymore. We saved ourselves a lot of time and memory. As we've seen, we can do our standard stuff just to remind us. If we have an RLE list, it is a list. So if I wanted to get the actual RLE vector from it, I can just do double square brackets. It's a list, remember. I say chromosome 12. I could have done $chromosome 12, no quotes. That would have worked too. I'm going to extract the region I know is interesting, the region around the gene. And then I'm going to create a data frame so we can subset RLE lists. Remember, just like standard vectors, even though they're these compressed objects which don't look like vectors, this RLE we can just subset like a standard vector. I've extracted the signal from it there, and then I create a data frame for ggplot. I give it the position. I will scale our signal a little bit, and then I can load ggplot. I can actually make now a representative coverage, what we would see in IGV, just doing it in standard ggplot, where we got our signal depth scaled because it's times by 100. Position, signal, and then just a geom line. Okay, does that look like it should do? I think we had the picture earlier on. It's hard to tell, I guess, right? Yeah, I guess. 1, 2, 3, 4, bumpy, bumpy. Cool. We can also, I mean, coverage accepts lots of different types of objects, and I think this is what makes it so flexible. So if you wanted to, you can actually read in the BAM file first with read GA alignments, and then once you have that GA alignments object, so that would be here, I can then run my coverage on that, and if I made a plot, it would look exactly the same. This coverage works for right from the BAM file, from the read in GA alignment object, which means I could just do a little bit more filtering, a little bit more manipulation before I make the coverage. But also, if I wanted to do really anything, right, I can actually use G ranges and G ranges lists, and I can make coverage from them too. Okay, so now we could take embed files and make coverage, create our own G ranges internally of some metric over some regions, and we can create something we can visualize in our coverage first, an hourly list, and then we can export that to visualize in IG. So if we wanted to turn, our reads here, we saw in our last session, we should be able to turn our alignments to G ranges, and then we can make a coverage just from that G ranges. Great. And then we'll get our coverage we can export. If, remembering, we have RNA-seq reads, and they have splices in them, junctions, we don't want to export it as a G ranges, because that means we're going to get the full range of where the read mapped. So if it had a gap in the middle, we're going to get from the very beginning to the very end. Whereas if we run it from a GRG list, it's going to split that read. And it's just going to give you the tiny bits of coverage on either side, tiny bits of the read on either side, and then we can run coverage on that. Great. I guess this is exactly what I was saying earlier on. If we wanted to, then it just means that we can just do things like I said, bigwig is strandless, right? It's only got 4 columns, but I could make a stranded bigwig, right? I could say this is my positive bigwig and this is my negative. I can overlay them in IGV. So to do that, I would just take my aligned reads, the ones I read in here. The ones I read here. And now I will get the strand, and I will just take those on the positive strand, make a coverage from them. Here I'm just, showing you chromosome 12, and then I'm just going to export that, just as we did the other session for when we read in an RLE and exported it back out. Now I can export this newly created RLE list from my BAM file and export it to a bigwig. Okay. A nice thing about coverage function is that it's going to allow for the specification of one really useful, probably more than one, but it's the one I really remember, useful additional parameter, weight. Okay. So the weight parameter is going to apply a scaling factor to the scores. So once it's worked out how many reads were at a position, it's going to apply this scaling factor, and then this is going to allow us to normalize our signal a little bit, not in a very complex way, but a very sensible common normalization is to say number of reads at a position per million reads which mapped to my genome. Okay. So here we can just apply a standard kind of weight. I'm just going to take my heart BAM and I'm going to apply a weight of 10. And if I look here, I can see that all the scores I've got are up by 10. Right, it's just times this, every value it got by 10 compared to the original. So if you can now combine this with what we used in the session earlier on, and I said about IDX stats BAM being really quick. It is fantastically quick. And in this case, I could use this just to take my BAM file, get me the total number of mapped reads right here. Once I know the total number of mapped reads, I could use that as my normalization factor. So in the next slide, we're going to take our sum of all the columns of that particular column. So there's only one value there. But if you had multiple chromosomes, you're going to want to know what mapped to the entire of your genome. And then I'm just going to run coverage on my BAM file and just do 1,000,000 divided by the total number of mapped reads. Okay, and then I can just do a normalization akin to reads per million here. Means if I had another BAM file, I could do the same normalization. I could apply this type of normalization. And when I open them in IGV, I don't need to worry so much that this was sequenced 20 times deeper. This had 100 times more reads. It's going to be normalized. So I should be able to just visualize them and compare them side by side. Right, so that's how we can do, really go from a BAM file to summarize the signal into a per base pair score. Okay, and that's what we would want to export now for bigwigs. I don't actually export it here, but or maybe I did a few slides ago. Got there, export. That's really useful for us both to visualize it. I told you that you can actually visualize a BAM in IGV, but it's slower. This is going to allow you just to generate your normalized bigwigs in a reasonably sensible way. We can use it to summarize and count the score and say peaks, but if we really want to do RNA-seq, is there a slide missing there? If we want to do things like RNA-seq, we're going to want to use something a little bit different. Okay, I think I've actually got a few more slides before I get there. But we can still get some sensible approximations from our bigwig at this point. What we can do here, last example, we're going to take our TXDB object, and I'm going to extract here our exons by for this particular genome, H38. I'm going to say give it by gene. And then I know that the SLC 25A3 gene is 5250 by Entrez ID. So I'm just going to grab it directly. And then this is the G ranges and the exon positions for this gene, right? We can already see a part of the problem of summarizing this here. This exon overlaps heavily with this exon. This exon here starts at the same position, ends at a different position. This one starts at the same position, starts at a different position, but ends at a different position, ends at the same position for the second, right? So these are overlapping exons in a complex multiple transcripts in a gene. Right? But we can take these G ranges for now. We can get our coverage from a BAM file, and then we can use, like we saw in our genomic scores lecture, we can use a G ranges to subset and give us the scores over every exon. Right, here are the mean scores over every exon. So we can do that really quickly in 2 lines, 3 lines, maybe a little bit more. Yeah. We can go and we can get sensible signal per exon for our RNA-seq BAM file. Okay, so that's pretty straightforward. But let's reiterate something there. This is going to work if I just care about signal, total signal in exons. But it gets more complex if we want to summarize all the signal which could belong to a gene, because as I showed here, these exons aren't unique. They overlap. They start at different positions, so they are actually different, but there are different splice variants for this gene. And in order to account for all this complexity in gene models, when we do our read counting, we need to be a little bit cleverer, or at least the programs we use are going to be a little bit cleverer. So we're going to take advantage of a specialist counting tool in Bioconductor, Summarize Overlaps. When we come to the RNA-seq, we're actually going to show you another one as well. So we're going to show you feature counts as well. So that's a very popular tool. We're going to use it from within the R. But Summarize Overlaps, I hope you'll see why it's so good to work with this inside the R framework. Both are popular. I think feature counts, because you can use it outside R, it's perhaps more popular. But most people, or like I showed you, these guys have written feature counts beautifully inside R too. So the really important thing about Summarize Overlaps, I've already alluded to, is it's going to be able to assign reads to meta features, genes rather than exons. So we're not going to worry that this read maps to 3 exons which all overlap each other. We're just going to say the read mapped to somewhere within the exons for that gene. Okay, and that's going to be great for differential gene expression. And most of the time, if you're doing this or single cell RNA-seq, you're concentrating on genes, not necessarily transcripts. It's also going to allow us to, or it's going to handle for us, reads overlapping multiple regions. So if we have a read which maps to our gene and perhaps another gene as well, it can be in both because they're on this different strands, but we've done strandless RNA-seq. We don't know which one our reads come from. This is all going to handle this for us. So it's just going to take care of this under the hood. If we can see, part of the problem here is this, that, we couldn't decide if we have all these reads here spanning these exons, we just want to count this once, right? We can't count this part in this exon and then summarize it up. Otherwise we'd get 2 times. And then we've got to decide these things which span into exons. So this read here, or this read here, there is no gene model which accommodates for an intronic read, a read mapping into this intron. Do we count it? Do we not? Summarize overlaps will take care of this for us. It has options to allow you to include these things if you needed to, but by default, it's going to do a great job. Like I said, often we don't care about this complexity of exons overlapping, maybe alternate exons here and subtle differences. We just care about, does a read land somewhere within the exons on this gene and only in that gene, so that I can trust that it's to do with that gene's expression overall. Maybe you're not so interested in this exon's expressed differently to this exon, yet. So when we use this summarize overlaps function, importantly, by default, a read can only be assigned to one feature. So this happens very little for standard RNA-seq, I think, in main mammalian genomes. But there are situations where you have a read which could be overlapping 2 features, or you have a feature, a gene within another gene. And it really can happen for when you start to do nuclear RNA-seq. You need to count the full length of a gene, not just the exons. So we need to keep this in mind, and it's worth noting here, a read can only be assigned to one feature. Right. Most importantly, when we start to do our exon level counting later on, we're really going to care about this. So as with every function in Bioconductor at this level, summarize overlaps is well integrated with other Bioconductor objects. So we'll often accept many different types of Bioconductor objects, which we should be familiar with, and just be able to give us a result out. So it can read counts from BAM or GRanges objects. So you could just say, I want to take my BAM file in its entirety. You might say, I'm only interested in particular reads, right? So there's something else I know about my reads. I'm going to go in and just grab the ones which had IDs from a particular library and count them. Right? So we have additional levels of control that we can not just use the raw BAM, but we can use our own GRanges too. And then the summarize overlap can count over GRanges. So these are going to be, intervals, peaks, exons, or most usefully for RNA-seq, meta features, GRanges lists, which are meta features genes. Okay, so to count non-overlapping regions, we can simply specify the path to our BAM and the GRanges of regions to count over. Right, so we're going to make this very simple for ourselves. We'll take the genes from our TXDB object for H. sapiens, so use genes, and it's going to give us the full range of the genes as a GRanges. We can then use our promoters function on this, and we're going to take 500 upstream and 500 downstream from the promoter, from the TSS, around our TSS. And now I'm going to run the summarize overlaps over these GRanges using my heart.bam file. Okay. It's going to give me back, I think, the last type of object we're going to see. It's a really beautiful object, a ranged summarized experiment object from the summarized experiment package. Okay. So this resulting summarized experiment object is going to contain the matrix So the kind of table of our counts over the regions of interest. Whatever we specified here is going to be in the rows. As well. Oh yeah, and in the columns then will be our samples. Okay, so we only have one sample here, heart.bam. So we're going to have a matrix, mostly rows, right? Rows for all the regions, and then one column. We can actually see that here if we look at the object. If we type this in, it says it's class range summarized experiment. It tells us the dimensions, 25,000 genes, so 25,000 rows, one sample, no metadata, has one assay, counts, right? So these are our counts of reads over these regions, has row names, which hopefully you can recognize now. This is our entrees gene IDs, 1, then it was 10, whatever. We have row data, names, so that's our gene IDs again. And then we have our col names, which was the name of our file, which we just used to process, right? So it's just taken the name from here and it's attached it as col names. If we want to actually get down to the matrix within our object, so we want to extract that, maybe write it to file, maybe do some of our own calculations. We just do assay on our summarized experiment object. You're going to use this assay a lot for single cell in bioconductor as well. Assay on this object, and it's going to pull out the first assay in our summarized experiment. Okay. We only have one here. So it's going to pull out our counts assay. And if we look, we can just look at the first row. And we have the counts over this gene for this particular file, 2,934. Right, so over the TSS for this gene, for this sample, this is how many counts we have. Really lovely thing about the ranged summarized experiment, because there is an equivalent summarized experiment. And that's just the matrix and some of these other things. But a ranged summarized experiment also contains the G ranges we actually counted over, right? So in the same order as the matrix, and we saw we had 110, 994, 997. It's actually got the TSS information here stored within the row ranges of this particular range summarized experiment object. Right? So to get these back as a G ranges, we just do row ranges on our summarized experiment, and we have the same G ranges we used to count. So this object's got a lot of the information. For gene models, remember, we often want to assign reads to each gene, and so we count all the reads overlapping any exons within a gene. Right? So in that case, we wouldn't be giving, we would be giving G ranges lists, right? So we're going to do here, we're going to go give our TXDB, on the right slide, sorry. Yeah. We're going to go get our TXDB for our Homo sapiens HG38. And rather than going exons or genes, we're going to go exons by. We're going to have exons by gene. And as we saw yesterday, this is now going to give us a G ranges list. I'm just going to look at the one for this gene of interest. But inside that G ranges list, we have all the exons associated to that gene, even if they're overlapping here. So this is cool because this means that we have the feature, sorry, the meta feature, the gene, and all the features which comprise it kind of contained together in this G ranges list. And then if I want to go and do summarize this for RNA-seq, I can run the same function, summarize overlaps. But now that it's a G ranges list, it's going to deal with it in the appropriate way for a gene. And it's going to give me the same file, but the same data back, this range summarized experiment. I have the same number of rows, I think, do I? Or maybe a little bit less. Less. I have still the number of rows for all the genes, right? I have one column. And I have the same row names, right? But actually the row data now, the row data changed a bit for me. And this time, because I counted over a G ranges list, I'm going to have a GRanges list stored in this row ranges element. Right, so now I have a similar assay object. We have a lot more reads mapping to this particular one, right? Because we're doing it with the whole gene. So I have the same assay object, and I have a very similar structure. It's still a range summarized experiment. But now if I go look at the row ranges, we have the information again we counted over. We were summarizing any reads landing in any of these exons for this gene now contained here. Okay. So the summarize overlaps, as you would hope, as you saw here, it gives us all these rows, but only has one column. Right? We hope then that maybe this will accept multiple files at a time. It does. Sorry, how am I doing for time? Very well. It does, and we can actually pass in to summarize overlaps. Here are exons grouped by gene, and we can now give it multiple BAM files. So we can give it here the two, the heart and the BAM, what we wanted to compare. It's then going to go do this counting and return back to us now a summarized experiment object with two columns. And now when I get the assay, I have very usefully my column names along here, my rows, and I can start to compare, maybe after normalization, the number of reads mapping to genes across columns pretty easily. We've got a few slides left. Great. So as with every one of these lectures we've done over the Bioconductor a few days, there's always this worry about memory. Right? I am running out of memory often if I'm doing it on my computer. If I'm on a nice bigger computer, if I'm online or AWS or something, maybe I can give myself more memory. But we need to make some sacrifices of speed versus memory. And especially if we're counting from many BAM files, we can actually configure this so that we can trade off our memory usage versus how quick it's going to run. Okay, so less RAM used, it's going to take longer, but maybe I can do some other things on my computer. To control the amount of memory used in these functions, the best way to do this is to actually create a new kind of wrapper object for our BAM file. It's actually called a BAM file object, and we have to use this BAM file function in order to specify. So we do BAM file, give it a path to the BAM we want to wrap up in this object, and we get this BAM file object from our samples. This means now that when we have this BAM file like here, we can actually specify particular parameters we want to attach to that BAM file. Right? So if this BAM is really big, maybe we want to change here the yield size. Okay, so yield size is the number of reads it's going to read in before it tries to do some counting, and then it's going to read in the next set of reads, and it will add the counts from the next set of reads to the current counts. And effectively, just like we're sorting, we keep this low. It's going to have to do it lots and lots of times. If we make this really big, it's going to read it all into memory. And once it's in memory, it's going to be able to do this quite fast. If you're on the HPC, if you're on a big computer, you don't need to worry about this. You might even set your yield size to bigger than the number of reads you have. Okay. In this case, it does make it very easy to control. Once we have this BAM file object constructed, we can just pass that to our, where we would normally put the path to the BAM, the my BAM, and it will do exactly the same thing, either slower or faster, depending on your yield size. Okay, so when counting multiple files, though, we probably want to use something like a BAM file list. And this is very similar. We just need to give it, rather than one file, we give it two files at a go. And then we give it a yield size here. And now as we pass in this BAM file list, it's going to go and read both these files. I probably am serial to start off with, but it's going to make sure it never reads in more than this number of reads. Okay. That's how we can go from our BAM files using our very simple things like exons by here. We can take our exon by straight from our TXDB HT38. As long as that was the genome we aligned to when we do the summary, we can get our little matrix of counts here. And it's this then, the summarized experiment, range summarized experiment object is the input for things like DSeq2, right? The DSeq2 object where we're going to do all our differential is just an extension of a summarized experiment object. So we're in good place now that we can do this counting. I got one more thing. I do have some exercises. I got one more thing I wouldn't mind just mentioning, but maybe I could share my R. I got to find my R, which isn't. So let's have a look at that summarize overlaps. Oh. Go load genomic ranges maybe. So hopefully I'm sharing my R. Summarize overlaps. Let's get rid of that. You can see it's got lots and lots of options here. One thing I haven't mentioned, and I actually can't see how we control it here. Maybe it's not really a big point because I think it will be handled under the hood. But one thing I didn't mention is parallelization in terms of using multiple cores in your computer. My understanding is summarize overlaps will. Sorry, I'm going to share my screen again. If I can find my studio. Summarize overlaps is built into Bioconductor, and they're going to take advantage of all the Bioconductor framework for using parallelization. Okay, and they are the place to go for this is BioC parallel if you really want to understand this a little bit more. So you can see it's one of the top ranked packages for Bioconductor. The reason we don't talk about it explicitly, and again, someone in this audience is going to prove me wrong because maybe it does work on our Jean-Nicolas. Sorry, won't work on Windows. But BioC parallel allows us, does it work on Windows? Yes. So BioC parallel really just allows you to use multiple cores. It has abstraction layers, which are going to allow you to run jobs across big HPC environments on different machines. It can talk through SSH ports. So BioC parallel, which handles all this, trying to use as much of your CPUs as possible. My understanding is for things like Mac and Linux, this is by default. It will try and use a certain number of cores on your machine. And you may find that when you start to run things like our summarize overlaps, you might find that you start to use a little bit of memory. And that's because even when you set this yield size, that yield size is per CPU per job. So if you ever have any issues with summarize overlaps, it's worth looking at BioC parallel as well and seeing how you might be able to turn off or even turn on using more CPUs in your computer, allow you to speed it up. I think we'll cover this a bit more when we come to the RNA-seq later on. So that's the end of the session for today. I'm going to hang around a little bit. We do have some exercises in the slides. If anyone wants to go through them or they want to go through, the actual solutions, I will be here until 4:30.